in international and national journals of high repute to his credit. He has research projects funded from DU, DST, and DRDO. So I welcome you, sir, to this international e-conference. I would like to invite and hand over the digital platform to Professor Surendra Singh, sir, and Dr. B.K. Singh, sir, to conduct this session. Over to you, sir, Professor Surendra Singh.
Thank you, Seema, for the kind introduction. And today our first speaker is Professor Kasturi Mukhopadhyay, and she is working as a professor in School of Environmental Sciences, JNU, New Delhi. She did her PhD from Saha Institute Nuclear Physics, Kolkata, and MSc and BSc in Chemistry from Jadavpur University, Kolkata. She then continued her postdoctoral research first at University of North Carolina at Chapel Hill, North Carolina, USA. Next at School of Life Sciences, JNU. And finally from LA Biomedical Research Institute at Harbor, UCLA, California, USA. And then she joined as associate professor at School of Environmental Sciences, JNU, New Delhi in 2006, and currently working as a professor. And the broad area of her research is environmental microbiology, focusing on antimicrobial agent development against drug resistance pathogens, their adaptive response and association with bioaerosol. She has supervised more than 20 PhD MPhil scholars and published more than 45 publications in high impact international journals with an H index higher than 30. And she has published book chapter and filed patent application from the outcome of her research. She has been editorial board member of Microbiology Scientific Report, that is Nature Publications, and along with her research work, she teaches MSc and MPhil courses at School of Environmental Sciences, JNU. So I'm welcoming Madam for this conference. So kindly go ahead.
Yeah. Am I audible?
Yes, Madam.
Yeah. Thank you. Thank you for nice introduction, Dr. Surendra Singh. And also I like to thank the organizer, particularly Dr. Umesh Kumar, for inviting me and for having me in this conference and allowing me to share my research work with all of you. And I like to share the screen first. Is it, can you see it?
Yes, ma'am. Yes, ma'am. Yes, ma'am.
Yeah. Yeah. So, you know, in my long journey from chemistry to microbiology, being a chemist, I landed up in the microbiology in my postdoctoral studies. And this microbiology actually kind of fascinates me in so many ways, particularly the power of the microscopic world. Now we see we are crossing through this COVID pandemic. Now we can understand that how much power in that tiny particle have, and they can actually make us non-living if they want. So in that context, today my title is the Antibiotic Resistance in Environment and Natural Therapeutics. So, you know, throughout the history, there's a continuous struggle between us and disease-causing microbes. But, and for that we develop antibiotics. We know all that kind of a history, that Alexander Fleming discovery of the penicillin, and it has saved millions of people during the World War II. But at the same time, we need to understand this microbial world actually staying with us for billions of years. We are just came to the earth. We kind of a conception stage, but they are living in this art for the moment this art has started. So when in the discovery, in the Nobel Prize lecture, Alexander Fleming actually warned us how to use the antibiotics, and where I will give you that kind of a essence, that where actually you are standing at present, and from where we can actually go and look at the scenario, and so that we can change ourselves. We need to actually understand the microbial world much more prominent way, much more other way. The way we actually understand that is not enough. So now we produce a lot of antibiotics. So how these antibiotics works? Either they inhibit the cell wall of the microbes, or they inhibit the DNA replication, or they inhibit the protein synthesis, or inhibit the different kind of enzymes. I hope you can see my slides. Actually, I'm moving my slides. Okay. Now, if we see the timelines of discovery of these novel antibiotics, that from 1930 to 1980 is a golden era of antibiotics. And in 1941, that penicillin came into the picture. Before that 1930, our journey started, and we have produced a lot of antibiotics, and we thought that we actually own over this infectious disease. We don't need to fear about the infectious disease anymore. And if you see that after 2000, there's a hardly we are producing the antibiotics. It's not only that we own over, but there are other reasons. The reason is that most important reason is the antibiotic resistance development. I want to see that Staphylococcus aureus history, because the antibiotic era has started from the Staphylococcus aureus, when the Dr. Alexander Fleming discovered penicillin. So in 1941, penicillin has been introduced in the clinic or in the market, and now we don't use penicillin anymore, because from 1950 it is totally resistant by the Staphylococcus aureus. And to combat that, in 1959, we produced methicillin, the same group of antibiotics, but within 2 years, the resistance appeared against the methicillin. And that is, I'm talking about MRSA today, because that is known as the methicillin-resistant Staphylococcus aureus, that is known as the superbug. To manage that, the vancomycin has been produced at the same part, but it's very toxic. It has been introduced to manage the MRSA infection. It has been introduced around 1999, but within 2-3 years, we saw the lot of cases of the vancomycin-resistant Staphylococcus aureus. In 2003, the FDA, after a long time, FDA approved daptomycin. Now daptomycin resistance, also Staphylococcus aureus has been also reported. So what to do? And this is a scenario of with all the antibiotics. Like, you name antibiotics, that is the penicillin, streptomycin, tetracycline, erythromycin, whatever. If you see the introduction here, and if you see the appearance of resistance. So now these bugs actually manage to resist all the antibiotics we are going to produce. Now, to produce one drug, it takes almost 20 years. Now, why the company will produce antibiotics anymore, when within after 20 years, a long struggle, we actually bring one antibiotics in the market, but then within 2 to 3 years, it actually doesn't work anymore. So antibiotics is a fading miracle? Yes, it is a fading miracle. We need to understand now. We have to go to the natural therapeutics. I like to discuss with that today. So now, if you see that development of resistance due to that long years of spending to develop one antibiotics in the market, and also the development of resistance, only 4 new classes of antibiotics, only for the gram-positive bacteria, not for the other bacteria, has been introduced in 2000, that linezolid, daptomycin, firamexin, and bedaquiline. And we already told that daptomycin resistant strain of the Staphylococcus aureus already has been reported. Now, how do these microbes resist antibiotics? They just produce enzymes which destroy the drug. They prevent the penetration of the drug. They alter the drug target site. and then they rapidly eject the drug. And I'm giving an example, like penicillin, the miracle drug in the World War II, but now they produce, the Staphylococcus aureus produce beta-lactamases that they produce per second 1 lakh lactamases, and each of them actually can able to kill 1,000 penicillin per second. Now you believe that in per seconds 100 million molecules of that penicillin drug destroyed. So penicillin is not active anymore. And then we have that, introduced methicillin. What they do, methicillin beta-lactamases cannot destroy methicillin, but what they did, they actually changed their penicillin binding protein and through the make gene, and then this methicillin cannot bind this newly penicillin binding protein. That is, they altered the drug target site. In that way, it is they have a very simple machinery, and their growth time is, generation time is 20 minutes. In that, few days or few years, they are able to resist all the drugs we produce. This is the examples. So now, what are the major problem of this antibiotic resistance? One is a major problem is that inappropriate antimicrobial use. In India, prescription not taken correctly, unnecessary antibiotic prescription, antibiotics for the viral infection, antibiotics without medical supervision, spread of resistance through different kind of a lack of hygiene and use of antibiotics in food. And in that era, actually in India, we are living. Another is very, very important. That is a horizontal gene transfer. That is, Staphylococcus aureus become a resistance, but it can transfer its resistance gene by several mechanisms to the beneficial organisms. We actually, in our whole body, the number of bacteria, beneficial bacteria, or number of bacteria is much more than the number of cells. Now, these beneficial bacteria, which were known to the beneficial bacteria, now maybe become more resistant. For example, one I'm talking about, the Staphylococcus epidermis, which actually coagulus negative staphylococci, and it is becoming now one of the rampant antibiotic resistance problem. And this resistance gene only not going to parent cell to daughter cell, but it can be transferred to the other species by simple conjugation, by the drug delivery to the viruses, by the transduction mechanism, and by the transformation that just by collecting from the environment. So there are two ways these antibiotic resistance phenomenon is happening. One is the inappropriate use of the antibiotics. Another is the gene transfer from one species to another species. And what is the scenario worldwide? If you see in worldwide, 7 lakhs people die each year because of the drug-resistant diseases as per WHO. This is the data of 2019. And if you see North America, even in Asia, even in Africa, you see that too many people are dying. But now the one problem, in this, because of this COVID case, because of the coronavirus infection, now it is actually whole attention has been taken towards that pandemic. And now I don't know how this infectious disease causing bacteria, how do we manage? Our attention should not be actually deflected from this viral infection, from the bacterial infection. It is an hour of need. You see 7 lakh people die each year because of the infectious disease, and India is one of them. Now India, if you see that now maximum antibiotics used in India, if you see, it is up to data is 2015. All the antibiotics produced by the whole world, it is India is kind of a dumping ground. We are actually maximum consumer of antibiotics in this world. And now there is one example I'm talking about, that is the beta-lactamases NDM1, that India, there's a news in 2014, started from 2010 to 2011, that India carries one of the largest burden of drug-resistant pathogens worldwide. And NDM1, which actually reported in 2008, names is New Delhi beta-lactamases. And so now people are actually looking at us. that how much antibiotic resistance bacteria we are producing. And if you see that already told that India is the largest burden of this multi-drug resistant bacteria. Not only that, it's the largest burden of the tuberculosis. We are sitting on a time bomb. Now tuberculosis are extra drug resistance. They are not even multi-drug resistant. Second line of drug is not working anymore. So now we don't know when it is going to explode. And also high resistance among the gram-negative and gram-positive bacteria. That is antibiotic resistance in the Klebsiella pneumonia, that actually named after that New Delhi strain, that is the M1 strain, and another is the antibiotic resistance in the Staphylococcus aureus. If you see that all the drugs available not active anymore against Klebsiella pneumonia, and if you see the Staphylococcus, in the case of Staphylococcus, except the linezolid, all available antibiotics actually are not working anymore. So that is the scenario. So now, there is establishment of antibiotic resistance surveillance in India, and the nodal center is in the Chandigarh PGIMR, and the AIMS New Delhi and ICMR is the kind of a coordination. And a lot of regional center has been actually developed to understand the main goals, to establish a network of hospitals to monitor trends of the antimicrobial susceptibility, profile of the clinically important bacteria and fungi, limit, which is actually very, very important for the human health. And also this surveillance includes comprehensive molecular studies to understand the transmission dynamics, so that we can understand the AMR much more. So a lot of work has been actually started in India. And one very important thing I'm telling that from the OS, in the OS, the gram-positive bacteria is more prominent, Staphylococcus aureus. But in this report in 2019, we're saying that, coagulase negative Staphylococcus aureus is one of them, that is the Staphylococcus epidermis, which are the beneficial for us. And also the kind of a gram-negative pathogen like E. coli, Klebsiella pneumonia, Actinobacter baumannii, and the Aeruginosa, Pseudomonas, they are all resistant to the third generation of the cephalosporin and the fluoroquinolones and carbapenem. We don't have antibiotic anymore to manage these kind of infectious diseases. We are actually running out of antibiotics, particularly in Indian scenario, because we are the hub, we are the generating the antibiotic resistance from all these bugs. You name tuberculosis, Microbacterium tuberculosis, you name the gram-negative bacteria, you name the gram-positive bacteria. We are actually producing, because of our inappropriate use, completely misuse of the antibiotics. That need to be understood, that need to be actually addressed properly. Now today I'm talking about that superbug that is actually rampant in your life, that is a methicillin-resistant Staphylococcus aureus. It was thought that it is a contact disease, but it's a kind of omnipresent. We contain a lot of Staphylococcus aureus in our nasal cavity and in our body system. But when our body immunocompromised or not working properly, then maybe Staphylococcus aureus become resistant. And methicillin-resistant Staphylococcus aureus is one of the most important nosocomial pathogen, means the hospital-acquired pathogen. Now, it is mandatory now, even in the Western hospitals, that the surgery room has to be free of MRSA. That's why the MRSA need to be monitored. And on top of that, MRSA now has been discovered that it is not only contact disease, it is present in the air, and that air need to be fully devoid of the MRSA infection, because then it will be very difficult to survival of the patient, which are the immunocompromised already, or the cancer patients. It will be very difficult if they get the infections due to the methicillin-resistant Staphylococcus aureus. It was a mostly healthcare associated infection, but now it is also community associated infection. It is lot of report is coming, and it's not only resistant to the methicillin, it is usually resistant to several other antibiotics. So Ammonium infection, which does, it causes skin and small skin and infection, but at the same time, it can actually causes the meningitis, pneumonia, all catheter-related infection, because it can form the biofilm. I'll come to that point. And also, it causes septic shock and a bacteremia. It's a very notorious pathogen. So our lab since 2006, when I joined, my lab concentrated on to understand Staphylococcus aureus and to develop antibacterial agents against Staphylococcus aureus. Now we started with the E. coli and also CONS, coagulative negative staphylococci, and we do with the plant-based and the host-based. And at the same time, when we get the all report of the presence of the staphylococci in the air, so one PhD students and two MPhil students, actually, I have given them objectives to understand the prevalence of Staphylococcus aureus in air and water and their resistance profile, and also the adaptive responses of the Staphylococcus aureus. So we are working in that 3 area continuously from last 16 years. Now, though I thought that I'm not going to talk with you, but I'm just sharing within 5 minutes that what about the prevalence of the staphylococci in the indoor bioaerosols. If we see, we have actually collected air samples in 2 different areas. One is the central laboratory of the animal sources, and another is a library, our P.R. Ambedkar Central Library of JNU. And we collected their samples through this active sampling procedure, and then we bring the samples. We then collected all the samples liquid, and then we have done the plating, and then we characterize them by various techniques like down staining, catalyst test, whatever test, so that presence of staphylococci or Staphylococcus aureus, and we then species identification by the multiplex PCR and the then determination of the resistance pattern. And resistance pattern we have done with the 14 conventional antibiotics, like all antibiotics of the different class, whether this is the cell wall biosynthesis inhibitor, or they are the remnant polarization agent, or the protein synthesis inhibitors. We actually take all the antibiotics from all of the classes. And what are the results? Our observation is that present our study highlights that presence of coagulase negative staphylococci mostly, but we got some Staphylococcus aureus strain and animal, and there are animal associated staphylococci and also human associated staphylococci in library and also in the animal house, but mostly around 50% are the multi-level resistance. Another most important observation that they are sensitive to ampicillin. We don't use ampicillin anymore. That means as they are not getting that selection pressure of that antibiotics, they are now becoming sensitive to the ampicillin. That is the only good point actually we got. That's the most important observation. If we don't use antibiotic for the longer time, then carrying a lot of gene is not easy for the bugs also. It's not easy for the microbes. So if they don't see the selection pressure of the antibiotics, then they actually release of those genes, and we got that observations. And we published this paper in the BMC Microbiology very recently. And then further, we started to understand that what is the air bacteria, what is the bacteria present in the air. And here, it's very difficult. I'm telling that only only less than 5% bacteria you can culture at the lab. So now the metagenomic study has appeared. By the metagenomic study, we can actually know, we can much better of the bacteria. And by the metagenomic study, we actually saw that in the air of that we collected from the central health, from our in the health university health center, and in the university we have seen that lot of proteobacteria, actinobacteria, and the firmicutes present there. But among them, though only low abundance, only 4%, lot of Pseudomonas, Actinobacter, and the Staphylococcus present in the air of the hospital environment. And we publish in the temporal data in the special session on the better air quality in the Journal of the Aerosol and the Air Quality. This is very recent in the 2020. We published both the paper on the prevalence of the bacteria in the air. So now coming to my own major work, that is the antibacterial agent discovery against Staphylococcus aureus mainly. And our approach is two approaches. One is a plant-based, another is a host-based. In this talk, in this next half an hour, I use the one word MIC. MIC means the minimum inhibitory concentration which is needed to arrest the growth of bacteria. So MIC actually, when we take a bacteria, we determine the MIC, and that MIC value tells us that how much concentration is needed to kill those bacteria or to arrest those bacteria. So MIC is low means the drug is good, and I am going to use that term MIC a lot in a couple of next few slides or next all of the slides. So in the plant-based, our approach was the curcumin. We all know the curcumin is a golden spice. It is a yellow-colored polyphenolic compound, and it is kind of a native to the Indian subcontinent and the Southeast Asia. And turmeric is a herbaceous flowering plant, and it is also known as the Indian saffron. We are taking curcumin every day at our home, and we use as a food additive spices and also for the textile dyeing, and it is an integral part of the Indian Ayurvedic medicine. And what is the it is the most important. So if you see the curcumin consists of the curcumin 1 and curcumin 2 and curcumin 3, and curcumin 1 is a major portion. And if you see that curcumin, there's a beta diketono moiety and there's a phenolic groups. And what they do is a lot of if you search the curcumin literature, you see that it does everything. That is anti-cancer. Mostly it has been established as anti-cancer, anti-Alzheimer recently, is a radioprotective, antiviral, antibacterial, antifungal, anti-inflammatory, anti-diabetic, antioxidant. You name, but till today, it has not established as a kind of a drug. Why? Because the major limitation of the curcumin is a low bioavailability. That is a tremendous poor aqueous solubility, the poor absorption, and it's a rapid metabolism in the body. So bioavailability is very less for the curcumin. That's why curcumin has not been came into the market as a drug. Though curcumin tablet, filling in the I have heard that filling into the capsule has been used in the Western country, but it's not approved by the FDA or any kind of agency as a drug. So because of that, so how to deal with that? So a lot of laboratory has been actually working on that. And here we have tried how to increase the solubility of the curcumin, our main approach. One of my students had given that project for his PhD topic, and he has tried 4 different ways. One is the curcumin in combination with the piperidine, encapsulation in the liposomes, and inclusion complex in the complex of curcumin in different cytodextrin, and metal complexation of the curcumin. These we have done in collaboration with the Professor Saurabh Das, Department of Chemistry, Yadavpur University, and we produce a lot of metal complex. I'm not going to discuss the metal complex today because of the time. And also we have actually in collaboration with Professor Dionis, our Department of Chemistry, University of Delhi, he's going to give a lecture tomorrow. And we have collected a lot of monocarbonyl carcinoids, which is actually modified by him in the central beta diketono part. So from him in the collaboration, we collected This is the he actually give a compound where he changed this diketone moiety to the monocarbonyl moiety, and then 66 compounds he has actually given to us. And among them, we have actually scanned them to the MIC value. And 33 compounds, we got a little MIC value, but among them, 4 compounds are very, very promising compounds. And these 4 compounds has a tremendous antibacterial activity. And you can, and they have actually rapturing the, it has a capability of the rapturing bacterial cell membrane. So what are these 4 compounds? This is the 4 compounds, that is 7D, 7I, 7N, and 7P has been named. And these compounds, if you see the value, they're all curcuminoids. And they are, if you see curcumin MIC is more than 256, but if you see this compound against methicillin sensitive Staphylococcus aureus or the methicillin resistant Staphylococcus aureus, and for the laboratory strain or ATCC strain or for the lot of clinical strains, you see they are giving a very promising results. The MIC value is very, very less, even at par with that oxacillin and at par with the vancomycin, which are the last resort of the drug to manage the MRSA infection. So next, he has actually done a lot of ex vivo efficacy, that you infect the tissue, and then whether we see that whether this compound could manage those tissue captured in the tissue, and we have seen that they can, as compared to vancomycin, rifampicin, and linezolid, our compound actually can wash off and can kill the bacteria associated with the tissue. We infected the tissue with the Staphylococcus aureus, and then we have added these drugs, these curcuminoids, and we got a very good results. And then preliminary, we have also started, examining in vivo model, and we have see that, if you see that 7I and 7P gives a better result compared to 7D and 7N, and so we have seen the investigation in mice infection model of the Staphylococcus aureus. Like, we infected mice, and we then, after the infection, we actually sacrificed those mice, and we cultured the kidney and the liver, and we have seen that these 7I and 7P actually can clear the infection if even in the mice infection model. But this is a very preliminary study. We need to work on that. And 2 paper has been published by Dr. Prince. Now he's Dr. Prince, and he's an assistant professor in the Magadha University. And in this 2020 and 2019, we got 2 papers. One is SS Omega, and one is the European Journal of Chemistry, based on these monocarbonyl curcuminoids, and with the collaboration with the Professor Rawat, both of the paper in collaboration with him. So this is about our curcumin story. And now coming to that post, in next 15 minutes, I'm talking about another post-derived peptides. One thing, we just try to imagine that we live in the microbial sea. Now, everywhere there's a microbes. Now, how do you survive? We have a very strong adaptive immunity. We produce a lot of antibodies, but we have one important thing is that innate immunity. Now, just you try to understand that how the insects flourishes without lymphocytes or antibodies. How a plant seed germinates successfully in the midst of the soil microbes. How our cornea of eye of animal is almost free of infection, always, and many more. What is the answer? That is our innate immunity. What is innate immunity? The major compounds of innate immunity is the antimicrobial peptide. These antimicrobial peptides produced by the host, by us, and all life forms produce, whether it's a frog, whether it is any kind of insects, whether bovine, pig, plant, us, we produce a lot of antimicrobial peptide. In our, we have a lot of periodic acetylcysteine and the defensin. And these are the peptides actually part of innate immunity. So what they do, they are also a lot of our immune cells contains those large family of the human antimicrobial peptides like acetylcysteine and the defensin. So what they do, these are the maybe how come they actually keep in this evolution, in this scale for the millions of years. They are actually our bugs, when they attack, they first face this antimicrobial peptide produced by the body, but they are not becoming resistant. Why? So maybe that this, if we try to understand the antimicrobial peptides a little bit more, then maybe the potential answer to the raging microbial resistance, maybe the antimicrobial peptides, and maybe that this is the future of the chemotherapeutic agents. So in 1980, this journey started by the Michael Jaslow, which is known as the father of the antimicrobial peptide, who actually first shown the meganin from the frog skin, which can manage antimicrobial, that this microbial problem in the frog skin. So from that, the journey then, for the last 40 years, lot of paper has been published to understand these antimicrobial peptides derived from various sources. And so what are they? They are actually 100 amino. They are very small, 100 amino acids long. They are peptides. They are cationic in nature, and they are amphipathic in nature, and they contain hydrophobic amino acids residue 50%, and they are membrane targeting, and they are broad spectrum antimicrobials. They are not a small spectrum. So one thing you try to understand, that is that they are cationic in nature. That's why they are told as a cationic antimicrobial peptide. And now, so what they do, they directly kill the microbes in the phagocytes, and they also indirectly do the immunomodulation. And actually, this antimicrobial peptide connects the immunomodulatory activity between the innate and adaptive immune system in the higher vertebrates among us. And so the fundamental problem, fundamental understanding, one thing is that if you see that bacterial membranes are generally more anionic nature, but our membranes, plants and mammals membranes, are more kind of neutral in nature. And these antimicrobial peptides are actually the cationic, and these cationic antimicrobial peptides, they are electrostatic interaction. They actually enter, and they by electrostatic interaction, they actually kill these microbes. It's just they capture the microbes. That's why the resistance are unlikely to develop in the AMPs, because these peptides target membranes, bacterial membranes, and resistance will be very costly solution for most of the microbial species. That's why for the billions of years, in spite of their continual presence, AMPs doesn't, no resistance has been developed against the antimicrobial peptides. So the lot of antimicrobial peptides are actually, I think I have 5-10 minutes time. Yeah. So a lot
Yes,
of antimicrobial. Yeah. Give me 5 minutes. I'm actually. So we're working on this alpha MSH antimicrobial. This is a cationic in nature, and it's adopts secondary structures, and it has a lot of play role in the immunomodulation. And if you see, this is a 13-mer peptide. Actually, we started working on the alpha MSH for last 16 years, and this is major thing is that it actually pigmentation, but we have shown by different peptides that it actually directly killing the microbes. So a lot of papers have published that it's highly effective in that Staphylococcus aureus. It is a C-terminal portion is actually active, and also not only that, it is active with even the E. coli, and also if we improve the cationic charges of this antimicrobial peptide, then the activity increases. in that way, and if you see, they actually completely destroy the bacterial membrane, like the gamicidin. And so this is the study. So what we did, we actually tried to develop a different kind of but the major limitation of this anti-alpha-amylase based peptide, that is they are inhibited by the biological media. So we started again rectify them by introducing lot of hydrophobicity, like arginine and tryptophan. And then first time we could manage to measure the MIC, because in the MIC experiment we need to use the microbiological media, and in first time we could manage, and this paper has been published in scientific reports. And then we have done a lot of modification. If you see, then I'm trying recent modification. 1 minute. This is basically, here is a cationic charge enhancement. This is the enhancement of the cationic charge and hydrophobicity. Then the stability, that's the issue on the stability. So improve the stability. We actually, introduce some kind of unnatural amino acids to improve the stability. And if you see in the complex media, first time we have shown that ANA9, ANA10, ANA11, these peptides based on the alpha message actually can give the MIC in a very manageable concentration. It is almost kind of a very important. You see, MIC is in the range of even 1 micromolar. So we have patented these 3 peptides very recently. And not only that, they have a very little cytotoxicity, and they have causes this killing of the Staphylococcus aureus from 8 log to 3 log, 5 log killing we have shown. And not only that, it actually inhibits the staphylococcal biofilm. If you see the staphylococcal cocci, is basically the major problem is they form the biofilm on the different kind of a catheter or different kind of implants. But these ANA9 and ANA10, these 2 peptides can actually inhibit this biofilm. And not only that, we have also done the in vitro cytotoxicity. We have done in vivo toxicity. They don't hamper the kidney or liver of the mice, and they also do no development of the resistance against them. And also their synergy, they're synergistically active with the oxacillin and the beta-lactam antibiotics. That's the most important observation. So what in the next new students, I have given the project to understand the combinatorial potential of these 2 peptides with the conventional antibiotics, so that we can revive them back also in vivo efficacy using systemic infection of the mice model. In India, so in summary, in India, antibiotic resistance is one of the major concerns. In Indian context, gram-negative bacteria is more challenging, and we need to understand innate immunity, and we need to more research need to be done for the developing antibacterials from natural compounds and the animal origin. So my last slides, my journey started with Dr. Madhuri. Now she's a scientist C, and then adaptive responses by Tatsina and the Poonam Tagira bios work. They are all assistant professors, and this is my present group. And Shwastik is working on the combinatorial potential, and also Sana did most of the work I reported by this Sana done by this biofilm work, and the development of the new and antimicrobial-based alpha MSH. And Prince has done all this calculation work. Kanchan is still working on the E. coli, and Josna has done all this cationic antibacterial peptide. And I also have a lot of collaborators, we got all the clinical samples from the Dr. Vinny Dhawan Ames, and Professor Diana Esad, I already told, Professor Saurabh Das, and Dr. Munna Sarkar, SINP. And also, I'm lucky to have a lot of funding from DBT, ICMR, SARB, PhD PARS, and the UGC. Thanks for listening my lecture. I'm now happy to answer any questions. Thank you so much.
Thank you, madam. It was very nice lecture, and you have shown very good presentation for the antibiotic development and resistance, and also use of curcumin as an antimicrobial agent. And in the corona time, so many people has consumed a lot of curcumin in the milk, because the curcumin is more soluble in fats than the water.
Yes.
So it was a very good lecture. And now if somebody have questions, they can ask to madam.
Maybe in the chat box. I can't see it. Yeah. I'm sorry, I have to dash because of the time constraint. I thought that I have time for the 3:15. I thought it's a 1 hour. But it's a 45 minutes. Then I realized later. Yeah.
I think there is nothing in chat box.
Yeah. If not, it's fine.
If there is no question, then we can present a momento to madam. Please.
Ma'am, please stop sharing of your screen.
Okay. Yeah.
Thank you, ma'am. Dr. Ashwini, please share the e-memento as token of appreciation to ma'am.
Thank you so much. So certificate, could you please send me by email?
Yes, ma'am, will do. Thank you, ma'am.
Yeah.
Thank you, ma'am.
Okay. Okay. Thank you so much. Now I'm leaving then. So I'm just.
Yeah, now it's time to invite the next speaker. But before inviting next speaker, I would like to really congratulate the team this Bandhu College Chemistry Department for organizing such a excellent program. If you see the brochure and if you see the name of the speakers, it's really, I can see the great efforts made by the team inviting across the globe, let's say, because I can see some people from the Canada, Professor Jha from the Canada. So it's a kind of international speakers are also here. So congratulations, team, for organizing such a nice conference. And thank you, organizing committee, for inviting me and Dr. Surendra Singh to chair this session. And now I would like to invite the next speaker of the session, that is Professor Bhumi Shankar, who has obtained his BSc in 96 and MSc in 98, and he did his PhD from IIT Kanpur in 2004, and then he moved for a short period in USA for a postdoc, and then he moved to the University of Liverpool for postdoc. Once again, the postdoc, it is called the Senior Research Associate position, and he stayed there for 3 years, starting from 2004, November 2004 to December 2007, and then come back in 2008 and joined the IIT Guwahati as an assistant professor, and then he moved to IISER Pune in 2010 and become an associate professor in 2014, and then full professor. We say like professor, not full, because we don't have this terminology, full professor or half professor. So become a professor in 2019. So from the profile, it is clear that he is a very prominent dynamic personality. He has received many awards, including a Star Award from the SIR and CRSI Award in 2022, and this is Star Award from SIR in 2021. So he has received many awards in his career. His research interests are mainly focused in synthesis and structure, physical property, and energy application of materials of inorganic, basically the inorganic particles or inorganic and material chemistry. So with this short introduction, I would like to welcome Professor Bhumi Shankar. Please, floor is yours, so you can start the session now.
Okay, thank you, Professor B.K. Singh, for your kind words of introduction. And I also thank Dr. Umesh Kumar for sending his kind invitation to give a talk on this very interesting conference that your college is organizing. And I also congratulate this Deshbandhu College for completing 100 years of existence, which is remarkable. And it clearly demonstrates the service that it has put forth for the country and for the people of India. Okay, so with these words, I will start my presentation now. Okay. Okay. So do you see my slide?
Yes, sir. It's
Yes.
visible.
All right.
Yes, perfect.
All right. Okay, so the title of my talk today is on a topic called hybrid organic inorganic ferroelectrics for piezoelectric energy harvesting applications. Okay, so you can see that this is some sort of a new class of compounds and application area that has came into prominence around 10 or 15 years ago, and the application domain of this particular class of compounds started only in the last 6 to 7 years. Okay, so this is our the main entrance to the campus. If you have not visited, please visit us, and also encourage your people to apply for PhD positions in our institute. Okay. Okay, before I go with my technical presentation, I would like to acknowledge the people who actually contributed to our research, particularly the students. Okay, this is the group photograph taken in 2021 May. Since then, a couple of students have left the group and a couple of more have joined, and we have not clicked one yet. Okay, so there are a lot of students who actually contributed to our work. I've given the names of the students who are either currently in the group or contributed only to this project, because I don't have space to write everybody's name here. And the work that I'm going to talk about was initiated by my second PhD student, Anand Srivastava from BHU, and then it was then taken over by Ashok Yadav, and later on people like Vijayakant, Sachin, and Swati have contributed to the work that I'm going to talk today. Okay. And I would like to thank a lot of collaborators, because this is a completely interdisciplinary area of research, and I do not have the enough expertise to address all the properties of our molecules. So I collaborate with people time to time to establish the materials properties of our compound. So their names are given here. And I also acknowledge funding bodies, especially the generous funding from ISO Pune from the beginning, DST India, SCRB India, and Armament Research Board. Okay, so these are the people who have majorly funded our research. Okay, so the moment you talk about ferroelectric and piezoelectric materials, one has to think about what is the basic property of all these materials. Okay, so when any material is called as ferroelectric or dielectric and so on, these materials are actually a fundamental class of compounds are called as dielectric materials. Okay, so dielectric materials are typically electrical insulators. Okay, so when we talk about conductors, semiconductors, and we know that there are insulators. Okay, so the very well-known example of an insulator is diamond. Okay, but then diamond is very hard to do anything with that. So there are thousands of molecules and materials which actually can change their properties under certain external stimuli. Okay, so those materials when they are insulators in the native state tend to polarize. Okay, during the application of an electric field, they are called as dielectric materials on whole. Okay, so what it means that the dipoles in the molecule will be randomly oriented in the absence of an electric field, and then when you place this material in between the two parallel plate capacitors and then apply an electric field, the dipoles of this molecule will tend to orient along one direction. Okay, or during the direction of the applied electric field. Okay, the positive end of the dipole will be towards the negative end of the capacitors, and the negative end of the dipole will be pointed towards the positive end of the capacitor. Okay, and we can see that these dipole the dipole moment of a material, which is termed as rho, is a product of Q and D. Okay, Q is nothing but your charge of the material, and then D is the space separated by the two capacitors, the capacitor plates. And then the dipole moment of a system then therefore will give rise to the polarization in the system, which is nothing but the polarization of a material is nothing but the summation of the dipole moment over a unit volume. Okay, so of course this equation becomes more complex as you deal with more complex phenomena, but for us to understand this particular presentation is that you can extract polarization from the dipole moment of the system. Okay, and the units of polarization is typically represented as coulombs per meter squared or microcoulombs per centimeter squared, depending upon the magnitude of the polarization the material exhibits. Okay, and you can also calculate the polarization of the system, okay, by computing the dipole moment of each of the molecule in the material. Okay, so that means that the number of molecules with the dipole moment of rho will give rise to the total polarization of the system. Okay, so these are all some physical relationships, so we don't bother too much about it. But what factors contribute polarization in your molecule or give rise to the dipole moment in the system? Okay, so there are largely 4 polarization mechanisms that are not discovered. The primitive or the basic one is nothing but an electrical polarization or electronic polarization, which comes because of the I would say that polarization of the electronic cloud. Okay, in the absence of an electric field, the electronic cloud is spherically charged, whereas during the application of an electric field, the electronic cloud is actually polarized and distorted in the shape. Okay, so therefore we can see that in the direction of the electric field, the positive charge of the dipole is created in the electronic cloud. Okay, the second important mechanism for polarization is the ionic polarizability. Okay, that means that the movement or the slight displacement of ions happens in the molecule during the application of an electric field. For example, if you have a cation and anion in the system, when you apply an electric field, they slightly move from their original position. Okay, so that can give rise to a polarization in the system. Okay, so we can see that one direction is now dominantly projected along the direction of the electric field. Okay, so this is typically with most ionic systems like sodium chloride and all sorts of salt materials will exhibit this polarization. So the third class of polarization is called as dipolar polarizability or molecular polarizability. Okay, so we know that as chemists that a lot of molecules are polar in nature. For example, HCl, water, and all sorts of molecules which have large electronegative difference between the constituent atoms exhibit permanent dipoles. Okay, so these molecules will exhibit a polarization which are called as dipolar polarizability. Okay, so in this dipolar polarizability, what happens is that the dipoles in these molecules change their orientation and tend to align themselves during the application of an electric field. Okay, so in the absence of an electric field, these molecules will have a random polarization or random orientation, and then once you apply an electric field, the dipoles orient along the direction of the applied electric field. Okay, so these are called as a dipolar polarizability. So if you compare the ionic polarizability and dipolar polarizability, the difference is in the ionic polarizability, the ions will migrate. Okay, the positive ions and negatively charged ions will migrate, whereas the dipolar polarizability, the entire molecule, shifts its orientation. Okay, so this is a migration or the displacement of the entire molecule in the system. Okay, so the final polarization is called as space charge or interfacial polarizability. Okay, so this space charge polarizability happens when the charge carriers at the electrode material interface migrates. Okay, so this typically happens because the electrical charges at the electrode materials interface will experience an enormous amount of an electric field during the application of an electric field. Okay, so because of that, the ions will tend to migrate very rapidly. Okay, so when this happens, and during the reversal of the electric field, the migrated charges will not go back to its original state. Okay, this is because they are actually trapped within the system. Okay, so this is because of the friction between the electrode and material interface, and because of the friction, these ions will not go back to its original state. Okay, as a result, these molecules will show an enormous amount of dielectric constant under the electric field. Okay, however, when you perform this electric field migration or the frequency of the electric field application, if you perform it at higher frequencies, the migrated charges will go back to its original state and remove the enormous dipole moment that is produced in the system. Okay, so that means that if you want to get rid of the space charge polarization, you have to perform this experiment at higher frequencies. Okay, which brings me to the next slide, that the dielectric constant of the material is therefore dependent on the frequency of the applied electric field. Okay, if you change the applied electric field frequency, the polarization mechanisms will vary. Okay, so for example, if you perform the experiment at very low frequencies, all the 4 polarization mechanisms will be prevalent. Okay, but then as you increase the electric field, one of the polarization will be reduced. For example, the interfacial polarization will go off. Okay, the space charge polarization. Okay, this is around 10 power minus 3 to minus 4 will be the frequency. Since I have plotted it in the log scale, you will see a positive log frequencies. Okay, so at lower frequencies, up to some threshold limit, all the 4 polarization mechanisms will be prevalent. But then when you increase the frequency of the measurement, the interfacial polarization will go off. Okay, so the next polarization which will go off is the dipolar polarization. Okay, so when you go to a frequency of about 10 power 8 log scale, then the dipolar polarization will go off, and then the ionic polarization will also go off at even higher frequencies, and finally only the electronic polarization will be present in the system. Okay, so the thing is that both this ionic polarization and electronic polarization will have a major contribution when the migration is very high. Okay, but when the migration is very low, their contributions will be very less. That means that if the electronic cloud is not distorted too much, the dipole moment in the system will be very low. Okay, so this is all we have to understand. Okay, so this is now enough physics for our system. So let's now go back to some chemistry. Okay, so since so far whatever we have discussed is a material with its dielectric constants or dielectric materials. Okay, so a material which is dielectric will exhibit a dielectric constant, and typically when you increase the magnitude of the applied electric field, the dielectric constant will increase in a linear fashion. Okay, that means that if you withdraw the applied electric field, the magnitude of the dielectric constant will come down linearly. Okay, so this is something like this. Okay, this is something like this. I hope that you will be able to see. So if you have plot the dielectric constant as frequency, it will linearly increase with the frequency, and then when it comes down, it will linearly come down. Okay, so this is called as linear dielectrics. Okay, however, there are a lot of materials which show nonlinear dependence for the applied electric field. Okay, so those materials are called as nonlinear dielectric materials, and these nonlinear dielectric materials will be of several classes. Okay, so the first class of compounds are called as piezoelectric materials. That means that when you take a dielectric material and you apply an external force, okay, if you give a stress to the system, the polarization will originate in this molecule. Okay, these are called as piezoelectric materials. Okay, therefore mechanical force induces polarization changes in the system. Okay, so the second class of compounds are called as pyroelectric materials, in which if you change the temperature of your measurement, the polarization changes. Okay, so that means the material actually is dependent upon thermal changes. Okay, so therefore they are called as pyroelectric materials. Okay, if these pyroelectric materials shows a reversibility of the polarization, that means that if you reverse the electric field, the polarization changes its direction. Okay, so that means that the polarization which is pointed upward in the first cycle of measurement will now point towards the lower direction, and that will be called as switchable polarization. Okay, and those materials are called as ferroelectric materials. Therefore, the electric field induces polarization reversal is experienced for ferroelectric materials. Okay, so the nonlinear dielectrics are therefore three classes: piezoelectric, pyroelectric, and ferroelectric. Okay, so if you see that what we have understood from so far is there are a larger class of materials which are called as dielectric materials. Okay, and piezoelectric will be a subset of the dielectric, and pyroelectric will be again a subset of piezoelectric, and ferroelectric is again a subset of pyroelectrics. Okay, and if you just go back to the structure of the molecules or the ions or salts or any material, and there's no crystallography actually determines the nature of their electrical properties. Okay, so what I mean by that is that if you look at the crystallographic tables, there are 32 crystallographic point groups, okay, which will contribute to 250 on plus crystallographic space groups. Okay, so all those materials with all those space groups can potentially exhibit dielectric properties. Okay, so if, for example, they all have to be insulated in the first place, then they can exhibit dielectric properties. Okay, so therefore any material will be dielectric, but not all materials are piezoelectric. Okay, so there are about 10 point groups of the 32 crystallographic point groups, or 11 of them, okay, are actually centrosymmetric. Okay, and because of that, the dipole moment in the system gets cancelled across the inversion center or the mirror plane. Okay, so therefore these materials will not have a permanent dipole moment in the system, and therefore will not show any nonlinear dielectric properties. Okay, so of the remaining 21 non-centrosymmetric systems, one has the highest symmetry. As a result, the dipole moment in the system gets cancelled even in that particular point group, which is called 432 point group. Okay, and then what is left over is again 20 point groups. Now all the 20 point group materials can exhibit piezoelectric properties. However, only 10 of them will have a unique polar axis, which will exhibit pyro and ferroelectric properties. Okay, so which means that as a synthetic chemist or a synthetic materials person, it is very challenging to design ferroelectric materials, because you should be little lucky or will have some extreme idea about controlling the symmetry of the system to be able to realize these properties. Okay, so with that background, let me now go to the ferroelectric material story, what we have here. And if you see that the commercial ferroelectric materials are potassium dihydrogen phosphate, sodium potassium tartrate, which is called the Rochelle salt, triglycerin sulfate, and there are a lot of ferroelectric copolymers such as polyvinylidene fluoride. These are all known. Okay, and of course the best among all are the ceramics of the perovskite family. For example, barium titanate, lead zirconate titanate, potassium niobate, and so on. Okay, and these materials have found applications in a lot of high technical applications, such as you have this ferroelectric random access memories, and then you will have infrared sensors, thermal switches, and then piezoelectric energy harvesters, which are called as micro actuators, and so on. Okay, so they have a lot of applications. Okay, apart from that, the recent application of these ferroelectric materials is their enhanced piezoelectric properties that can be utilized for energy harvesting applications. Okay, so what you do by that is that if you actually subject a stress on the piezo or ferroelectric materials that you have in hand, they will exhibit electrical signals, and you can harvest those electrical signals as voltages and store them in the form of capacitors or in the batteries. Okay, and since the piezoelectric coefficient of a material, which is called as the D33 units, is directly proportional to the remnant polarization of the system, any material with the high remnant polarization or remnant ferroelectric polarization will potentially exhibit high energy harvesting properties. Okay, so with this background, people have worked on this topic. The first discovery of piezoelectric energy harvesting, which are called as nano generators, are discovered in the year 2006 by a person called Zhongling Wang at Georgia Tech. Since then, there are a lot of design aspects have come into picture. That the first discovery was, growing zinc oxide nanowires and touching them with the tip of a AFM probe, okay, which actually generated small currents and voltages, which are called as nano generators. However, people have now tried to commercialize this particular nano generators by changing various design aspects. Okay, so the design aspect that we have adopted is the composite type. Okay, so I will explain those things when we talk about our results. Okay, so over the years there are so many materials that are utilized for this particular applications. Okay, people have used traditional ceramics, people have used organic materials, and people have used organic inorganic hybrids. Okay, and the traditional materials, although they are very successful, they have a lot of disadvantages. Okay, for example, they are actually very brittle because of their ceramic nature, and because they have very high melting points, their processing will be very cumbersome, because to bring them into the form of devices, you have to process them at very high temperatures and so on. Okay, also they have a very toxic heavy metal ions such as lead in the system, so that makes their application in wearable electronics very complicated. Okay, so for that reason, people have started working on organic materials and organic inorganic hybrid materials for energy harvesting application. Especially people are using these ferroelectric materials for energy harvesting applications. Okay, so here we see that a review article that we recently written on this particular organic and organic inorganic hybrids for energy harvesting, and we described their various applications and their advantages and applications. Okay, so I would recommend, if you're interested, to go into this review and have a look at it. Okay, so the typical ferroelectric materials, as I told, are Rockall salt, which is sodium potassium tartrate and potassium dihydrogen phosphate. However, the ceramics are actually dominant in this particular field, and because they are typically very robust. So a ferroelectric loop looks like this. Okay, so when you give an external electric field, the material gets polarized and gets saturated at a critical electric field, which is called as a coercive field. Okay, so this is called a saturation. No, this is not a coercive field. This gets saturated, and the polarization gets saturated, which is called as a saturation polarization at a particular electric field. Okay, so in that particular saturation polarization, all the dipoles in the system are oriented in one direction. Okay, however, if you withdraw the electric field, not all the polarization goes back to its original state. Okay, most of them will be retained in the original state, which is called as a remnant polarization. Okay, so remnant polarization is a polarization which is realized at the zero electric field. Okay, so if you want to then push the polarization into opposite direction, you have to again give an extra electric field in the opposite direction, and then the polarization gets reversed and gets saturated in the opposite direction, and the cycle gets completed by the forward and reverse electric field applications. Okay, so there are so many materials that are known, as I told you, the ceramics, but then recently people started working on small molecule based ferroelectrics. These are my some favorite examples, and this is croconic acid, okay, which exhibits a very high polarization of 21 microcoulombs per centimeter squared, which is very close to the polarization of barium titanate. Okay, so the proton tautomerism was found to be responsible for the reversal of polarization in the systems. Okay, so then people worked on simple acid-base co-crystals. Okay, so this is like a neutral acid-base co-crystal of phenazine and chlorinic acid. Here also the proton migration was found to be responsible for the ferroelectric polarization. Okay, because the proton will switch between the two states, giving rise to the two polarization directions. Okay, then people worked on simple organic salts, which is dominant in ionic mechanism. Okay, so this is the Dielsopropyl ammonium salts, which exhibits high temperature polarizations. Okay, so we can see that at 422 Kelvin it exhibits a ferroelectric polarization. Okay, and we can also prepare organic inorganic hybrids. This is an ammonium cation, and this is an inorganic halide chain. This is a manganese chloride chain, and this molecule exhibits a ferroelectric ordered state at room temperature, and at high temperature it exhibits a paraelectric state, which means that the polarization becomes linear at certain higher temperatures. Okay, so this material exhibits a very good ferroelectric polarization. The red graph over here is nothing but the leakage current. Okay, so the leakage current will show almost zero leakage at all the measured voltages. However, it will show a peak at the coercive field because the domain switching happens at the field, which gives rise to a peak at the coercive field. Okay, so the signature P loop and the JE hysteresis or JE curves are the signatures of the ferroelectric property of a material. Okay, and this material also shows an additional piezoelectric property. Okay, the D33 values, which is a piezoelectric coefficient, which is close to barium titanate and lead zirconate titanate. Okay, which is very rare for small molecule based systems. So as a result, these studies prominently, indicate the potential of organic inorganic hybrids and organic molecules for technological applications. Okay, so our group focuses on the heavier congeners of ammonium salts, which are called as phosphonium compounds, and we prepare phosphonium salts with simple organic substituents or organoamino substituents. The choice of amino group is because of their ability to actually hydrogen bond with the counter anions and increase the dipole strength. Okay, and in order to reduce the symmetry of the system, we try to judiciously make them heteroleptic, and we can actually what we can do is we can substitute one of the amino groups with the simple phenyl group or an organic group. Okay, or you can take an all organic system and substitute them with the amino group. Okay, in all the cases, the symmetry of the system gets reduced from tetrahedral to C3V to C2V, and as a result, the symmetry of the system can be lowered, which can potentially give rise to non-centrosymmetric molecules with ferroelectric applications. Okay, we also made a lot of ferroelectric metallurgic materials, okay, which are called MOFs, using pyridyl functionalized phosphorus ligands, but I'm not going to talk about them in this particular talk. Okay, so in our approach, as I told you that we have played around with the aminophosphonium scaffolds. Okay, the first success came when we substituted when we prepared this diisopropyl amino phosphonium salt with 2 phenyl groups. Okay, so this is called diphenyl diisopropyl aminophosphonium compound. This can be prepared by a small synthetic procedure like this. You start with the phosphine with the 2 phenyl groups and 1 chlorine atom, and you oxidize this with bromine and substitute this with the primary amine group, and eventually do an anion exchange, because the monoatomic anions were found to exhibit very low polarization attributes. Okay, so we wanted to have a polyatomic anion, and this PF6 salt of this diisopropyl diphenyl phosphonium compound was found to be ferroelectric along the polar B axis. Okay, because the molecule has a non-centrosymmetric symmetry, space group CC, and the second C, small c, is along the B axis, which is a polar axis. Okay, so which will show a typical ferroelectric P loop like this, and a J curve with the peak at the quasi field. Okay, and we also subsequently made a composite material of this in PDMS polymer and made a lab scale device and use them for energy harvesting applications. Okay, I will talk about the applications of this material in the subsequent slides. But then what we have to understand is that when we had this diphenyl and diisopropyl system, the PF6 salt was non-centrosymmetric. But for the other cations like the phenyl triisopropyl or triphenyl isopropyl phosphonium compounds, the PF6 salt was found to exhibit centrosymmetric space groups. This is because the PF6 is octahedral and it has an inversion center, and therefore the structure dominating force was played by the PF6 salt instead of the cationic phosphorus compound. Okay, so which means that the higher symmetry of the anion is not very good for most of the phosphonium salts. So we reduce the symmetry of the system, especially for the anions, from octahedral to tetrahedral, and in combination with various phosphonium cations, we were able to make 12 phosphonium salts, of which 8 of them were found to exhibit non-centrosymmetric space groups suitable for ferroelectric and or piezoelectric properties. Okay, and 4 of them were found to exhibit a very good the ferroelectric properties. Okay, and of which 3 of them shows very high polarization attributes. Okay, which is around 35 microcoulombs or 21 microcoulombs and 25 microcoulombs and so on. Okay, one of them, although is ferroelectric, shows a very low ferroelectric polarization, which made us to look into their crystal structure. So of all the diphenyl diisopropyl system, it forms a 1D hydrogen bonded assembly mediated by strong classical hydrogen bonding interactions along the polar B axis. Okay, whereas in case of this molecule, which has 3 phenyls and 1 amino group, it cannot propagate with strong interactions along the polar axis. Okay, so there is a strong NHF interaction is present in this molecule, which is forming a dimeric structure. However, these dimers are actually subsequently connected by non-classical CHF interactions. Okay, so which means that the dipoles are separated at a farther distance, and the long range ordering of these dipoles are not as strong as the other molecules. Okay, as a result, the molecules this molecule shows a very low ferroelectric polarization. Okay, And then subsequently, we made a polymer composite. And in this instance, we used a TPU polymer instead of a PDMS polymer. And this is the typical procedure with which you can actually make a polymer composite, which basically is mixing the solutions of the polymer and your compound, dissolving it, and then pouring it onto a glass mold, allowing the crystallites to grow into the polymer, and then you can take the polymer composite out for your measurements. Okay, so by peeling off. Okay, so now these composites can be placed between the two aluminum contacts or the copper contacts, and then you can subject them for mechanical energy harvesting application. Okay, so what you can do here is you can do a you can hit these devices with the external mechanical force. Okay, like this. So when you hit, the voltages and currents are produced in the system, which can be monitored with the help of an external connected oscilloscope. Okay, the oscilloscope gives you the measure of the currents and voltages that are produced. Okay, and you can see that typically these kind of signals are generated in thousands of numbers. Okay, so what happens in the system is when you actually hit the molecule with an external stress, okay, a piezo potential is generated at this particular surface, which allows the negatively charged electrons to be produced at the interfaces. Okay, so because of the higher electronic cloud, these electrons are actually now passing through the material to the opposite electrode, which is positively charged. Okay, so there is a movement of electron takes place from the top to bottom while you are hitting the molecule or if hitting the composite material. Okay, so this gives rise to a signal in the positive direction. Okay, the moment you withdraw the force, now the piezo potential is now removed from this surface. The electron has to now go back in the opposite direction, which will generate a signal in the opposite direction. Okay, so this is the signal in the opposite direction. Okay, so by means of this continuous impacting and release, you can generate a number of signal output voltages. Okay, and these voltages can be then stored if you want. Okay, and you can also then measure the currents that are produced during these measurements by connecting a resistor across this particular measurement, and you can measure the currents. So therefore you can see that the voltages and currents are generated during this particular mechanical stress. Okay, and now we can prepare composites with various weight percentage compositions of the ferroelectric material, and we will see that as we increase them in the loading of the ferroelectric particles, the mechanical properties or the energy harvesting properties increases up to certain limit. Okay, beyond which the performance is lowered. Okay, and this is because that if you crowd the composite with a lot of ferroelectric particles, okay, then there is a chance that the particles will be crystallized in your system with different directions of dipole. Okay, so therefore there will be a randomization of the dipoles present in the system, which will reduce the performance of the material. Okay, so therefore you need to always optimize your composition before any measurements. Okay, so we can see that as we measure the different materials with different polarization values, different output voltages are generated. Okay, so a material with the ferroelectric property gives higher output voltages comparison with the material with only piezoelectric properties. And when we change from one anion to the other anion, there is also a change in the polarization as well as the change in the output voltage. Okay, so you can also therefore calculate the power densities and current densities in the system, which will tell the efficiency of the materials. Okay, so these are all plotted for comparison, and we see that the best performance was given by the material with the highest ferroelectric polarization. Okay, which is the diphenyl diisopropyl ammonium salt of the BF4 salt. Okay, so this tells you that qualitatively that the changing the ferroelectric remnant polarization changes the energy harvesting properties of the material. Okay, so this is one observation that we have made in this particular contribution. Okay, however, what you see is that the voltages that are generated are very low because the molecule is organic in nature. So in order to increase the performance, we changed from an organic salt to a organic inorganic hybrid salt, and we used a simple phosphonium salt which has a phenyl and methyl substituents, which helps in reducing the symmetry of the system. And an inorganic component or the anionic component comes from the nickel isothiocyanate coordination complex. Okay, so this molecule again crystallizes in the non-centrosymmetric space group. It shows a ferroelectric polarization and JE curves, typical for a ferroelectric material. And then we made a flexible composites of that. Okay, we can also visualize the location of the ferroelectric particles in the polymer matrix using 3D X-ray tomography techniques. Okay, so 3D X-ray tomography is similar to the scanning electron microscopy, except for the fact that the probe in this case is not electron beam, but it is actually an X-ray. Okay, so this becomes a soft technique to an SEM technique, so that the polymer will not degrade during the measurement. Okay, so that will give rise to a precise measurement of the nature of the particles which are embedded on the polymer matrix. Okay, so I have actually blanked the polymer background so that only the ferroelectric particles are visible. Okay, so as we see that as you increase from the 15 weight% to 20 weight% of the material, the agglomeration happens. Okay, which is not good for your performance of the material. Okay, then you can also measure the crystallinity of the composite with powder X-ray techniques. We can also determine the characteristic of this particular composites using Raman technique, where the peaks due to the polymer as well as compound are present in all the composites. Okay, and we can also therefore test the viscoelastic or the stress strain relationship of the material using viscoelastic, using a mechanical force technique. Okay, we can actually subjecting them to a dynamic mechanical analyzer. Okay, so this is called a viscoelastic measurements, and the epsilon prime is nothing but your elasticity of the material. Okay, epsilon double prime will tell you the viscosity of the material, which I have not shown here. So as you stretch the material for different seconds, there is no change in the strain in the system observed. Okay, however, they can also show a higher strain at higher loadings, which means that the material is more robust than the simple neat unloaded polymer itself. Okay, so the best performing polymer can also be stretched up to 30% strain, which means that you go to 300%. 300% of the strain, the material is very robust, which means that the composite does not reduce, give up its elasticity even in the even you have a crystalline particle which are loaded in your system. Okay, so then again we measure the mechanical energy harvesting properties of the material. The 15 weight% material shows the best performance. This is the current characteristics, so which gives an output short circuit current of about 12 microamperes, which is very high. And you can also subject them to do this measurement under various load resistances, because for practical applications the material will actually face some resistance from the external surface. So as a result, you have to give a resistance in terms of current. Okay, so the 4 mega ohm is the ideal resistance for our measurement. Okay, and as I told you that the performance of the material varies with the load percentages. Okay, so we see that the neat TPU polymer is actually non-dielectric. Okay, however, when you load them with the ferroelectric particles, the dielectric constant of the material increases, especially at lower frequencies, where the performance of the ferroelectric particles are more dominant than the bulk polymer. Okay, so as you increase the loading, the dielectric constant of the system increases up to 15 weight%, and when you go back to the 20 weight%, the dielectric constant decreases, which means that the dipole moment of the system is reduced beyond the threshold loading. Okay, which tells you that the best performance of the 15 weight% material comes from the highest dipole moment of the system. Okay, so we can also then use the output voltages that are generated in your from your system to store it. Okay, so you can store it in a capacitor. Here we have shown a capacitor, and we used various capacitors with different capacitance values. And we see that by subjecting them to an external mechanical force, the capacitors can be stored within 60 seconds. Okay, the maximum storage points are reached within 60 seconds for all the capacitors, and the charge that can be stored in the capacitors can be plotted from the voltage relationships. Okay, V is equal to Q is equal to CV relationship. Okay, Q is nothing but the charge, C is the capacitance, and V is the voltage. And from the output voltage or the stored voltage, we can actually calculate the measured charges. Okay, so all the system shows a very good charging of the capacitors, and we then try to use the potential of this system for various other anionic counterparts. Okay, so we use the same cation and use different anions. In this case, it's a copper salt, whereas in this case, it's a bismuth salt. Okay, so in all the cases, we were able to see very good performance. Okay, we can also establish the non-central symmetry of the system by measuring the nonlinear optical properties of the system. Okay, which gives the characteristic null signature peak at 400 nanometers when you pump the laser with 800 nanometers. Okay, so these are the crystal structures of these various non-centrosymmetric molecules. All these materials are actually, ferroelectrically active. Okay, the dielectric constant of the system is unaffected, which tells you that these materials are actually does not have any ferroelectric to paraelectric transitions. And in some cases, we can also visualize the piezoelectric domains using piezo-responsive force technique, which is nothing but a special AFM technique. Okay, so in the AFM technique, you can actually subject them to subject the molecule to an external electric field, and then you can visualize the domains. Okay, so the dark and bright domains indicate the domains with different polarization orientations. Okay, so this is like one orientation, and this will be an opposite orientation. Okay, and we can also see that if a material is piezoelectric, the stress will generate a displacement of the material. Okay, so that means that if you give an external force, the molecule grows in direction. Okay, so this is nothing but the growing direction. You see that from the origin, the molecule is displaced to 350 picometers. Okay, similarly, from the origin, it can go to minus 320 picometers. Okay, so which can grow in the opposite and the negative direction from the origin. Okay, because of the external electric field, which confirms the piezoelectricity of the system. In fact, we can visualize the piezoelectricity of the system. Okay, we can also use a tip of the PFM to actually visualize the ferroelectric and piezoelectric properties. For example, you can record the phase changes. Okay, so this shows a 180-degree phase change. Okay, which means that the signals are actually flipped. Okay, so for example, if the polarization at this point is like this point it will be like this. Okay, and the butterfly shaped loop is characteristic of a piezoelectric material. Okay, so that comes from the amplitude changes, and this is nothing but a phase change. Okay, so the phase change, these two graphs are related to each other, whereas these two graphs are related to each other. Okay, so this is at a single point of this strip, whereas you are able to visualize about 5 micrometers of the surface in this particular measurement. Okay, so then you can also subject them to an external mechanical force. You can see the dipole aggregation using dielectric constant measurements. You can subject them to an external energy harvesting application, and the output voltages generated from these devices can be stored in a capacitor and so on. Okay, so all these things are again similar to what we have done earlier. And we also investigated some special ammonium compounds to generate some special new type of perovskite materials. In this case, we used an ammonium cation with the metal coordinating functions. This gives rise to a slightly modified perovskite structure. Okay, in the normal perovskite, the cadmium centers are actually connected by 3 chloride bridges on a phase sharing fashion, whereas in this case, the cadmium centers are connected by 2 chloride ions in an edge sharing fashion. Okay, the third chloride ion is terminal, and then is hydrogen bonded to the hydroxyl group of the ammonium cation. Okay, so this molecule goes from the modified perovskite to a normal perovskite at higher temperatures, because the coordination is lost at higher temperatures. Okay, so this we have mapped crystallographically, and we can also see the different phases present in the material using dielectric differential scanning calorimetry. Okay, so when it goes from 293 to 363 coordinated to the non-coordinated state, you can see that the space group does not change. Therefore, the molecule will be ferroelectric at both the temperatures. But at higher temperature of 400 Kelvin, the molecule becomes centrosymmetric. That means that it is going from the ferroelectric to the paraelectric phase. Okay, so we also measured the polarization using PE loop analysis, which gives a polarization value of 18.1 microcoulombs per centimeter squared, or 17.5. So we can also calculate the polarization using a point charge model. So in the coordinated state, the calculated polarization is 18 microcoulombs, whereas in the uncoordinated state, the calculated polarization is only 4.4 microcoulombs, which tells you that the modified ferroelectric structure designed by our cations is actually giving rise to a long range polar order. Okay, so that means that you need.
Sir, Bhumishankar sir,
I'm finishing.
Very sorry to interrupt you, sir.
Yeah, I'm done.
Time is exceeding. Actually, we have another.
Should I stop it here, or should I stop it here, or should I put a concluding slide?
Sir, can you please compile, since we have another session also.
I know. I understand. Give me a second.
Please,
no, I'll stop it here if you interrupt right away. No. Or should we conclude?
Sir, you can conclude,
You can
please.
conclude, please.
Yes, please. Okay, so we have seen many such instances, and to conclude that we have generated a lot of materials which are composite materials, which gave rise to very high output voltages, current densities, and power densities. And these materials are comparable to most of the composites that have been synthesized over the years, and our materials are in some instances superior as well. Okay, so with that I conclude and say that we have prepared a lot of organic and inorganic hybrids, and this can be used for ferroelectric energy harvesting and storage applications. Okay, thank you all for your kind attention, and I'm sorry for overshooting. You should have alerted me at least 5 minutes before. Thank you.
It's fine. It's not that late. But anyway, it was really excellent talk, and we are running out of the time. So, but anyway, there could be one to quick question if there is, please.
No. I think there's a session. We'll put it in the chat box. I'll talk to people. All right.
Yeah. There is one query from Seema. Just one question you can take out. So how we can improve the environmental compatibility of traditional piezoelectric material as they contain some heavy metals?
The environmental compatibility of traditional material as they contain some heavy metals. That is why we are trying to address new materials.
Yeah, okay.
That is why we are preparing new materials, because there is an inevitable problem with the ceramics. We are now able to actually prepare new materials which can be useful for practical applications, at least for miniature electronics. Okay. Thank you.
Thank you very much. If someone have something, they can directly contact Professor Bhumi Shankar. Everyone has this contact detail, so it can be easy for us to conclude now. So I will request Seema and Ashwin to proceed the session and offer him the token of appreciation and certificate.
Yeah, please quickly do it because it's the next session is due. Thank you.
Thank you, sir.
Yeah.
Congratulations.
Yeah.
Yes.
All right. Good luck with your poster sessions. Okay. I got.
Thank you, sir.
Now.
Thank you.
Yeah.
Now, I would like to pay my sincere thanks to both the invited speakers, Professor Kasturi Mukhopadhyay ma'am and Professor Ramamurthy Bhumi Shankar sir, for such an interesting, thought-provoking and useful lectures. I extend my sincere thanks to our chairpersons, Professor Surendra Singh sir and Dr. B.K. Singh sir, for conducting this session and sparing their valuable time for us. I request Dr. Ashwini to please share the token of appreciation for both the chairpersons. Thank you, Ashwini, and congratulations, sir.
Thank you, Ashwini, and all the best and good luck for the next session and the coming days.
Thank you, sir.
Namaste, sir. Namaste.
Last but not the least, I would like to thank all the respected members of organizing committee and esteemed participants for joining and making this session a successful event. Kindly note all the presenters are requested to join their respective Google Meet for our next oral and poster presentation session. Schedule for the oral and poster presentation session will be posted in chat box, and I request Dr. Bharti to please do the needful. Results of oral and poster presentations will be announced tomorrow during valedictory session. All the participants are requested to please fill the feedback form for securing their e-certificates. Feedback link has already been posted in the chat box. So with these concluding remarks, we are closing this session. Thanks again for your cooperation and patience. Thanks a lot. Thank you, sir.
Yeah, thank you very much.
See you.
Should I close the meeting then?
Yes, you can.
Thank you, Seema.